Hey science fans! Mucked up my last video, let's try again.
So my bumper DNA video was too big, so I've had to split it up. So for trilogy students, we're going into B1 in the cells, the structure of DNA, and for triple we're looking at the structure of nucleotides as well and how they link to being monomers and polymers. And then I'll have to do another one. Soz.
You definitely, if you get asked a question about DNA, particularly its structure, you need to know 3 words. Have I forgotten what those 3 words are? No, of course I haven't. It's a double helix polymer, which means 2 spirals of repeating units made of monomers called nucleotides. Probably can draw it better than I can say it. Let's go. Okay, for combined science and triple, I suppose, you're going to need to know the structure. As I already said, it is a double helix polymer. So the double helix means 2 spirals, okay? And this is made of sugar and phosphate, so we call it the sugar phosphate backbone. You need to know more detail, which I'll go into for the triple. And then this section in the middle is made up of complementary base pairs. Now, complementary means comes with. Okay, so the 4 pairs that you have are A, T, C, and G. And so if we zoom in, we might have C here, which means that would be a G, and A here, that would be a T. And they come in any order, really. And we just know that whatever the order is on one side, the other side is going to be the complementary opposing order, like this. Okay, now the order of the bases gives us a code to make a protein. So when we're talking about genes, they are a selection of DNA that codes for proteins. Okay, and if we get the wrong order, or we get an insertion, so we get like another letter just kind of whacked in there, that's when we get a mutation, which most of them we never see. They kind of get drowned out by all of the other bases that are there. But that's the general structure of DNA. So we've got the sugar phosphate backbone and complementary base. I just want to talk about where we find DNA. So we find DNA in eukaryotic and prokaryotic cells. So we're just going to talk about the difference between those. So these are both eukaryotic cells. These are both our nucleuses. And if we zoom into the nucleus, we have these structures in the nucleus called chromosomes. And if you zoom in on a chromosome, they normally come in pairs. And if you zoom in on a chromosome, they are made up of the DNA all coiled up in this nice uniform manner. And so if we zoom in further, that's where we see our DNA. And a section of DNA that you find on a chromosome is called a gene, which codes for proteins. Now, when we get into inheritance, we will talk about the fact that you have a pair of genes for each protein. One comes from mummy, one comes from daddy, and they are called alleles. And those are where we talk about characteristics and inheritance. But essentially, DNA is a coil of your sugar phosphate backbone with the bases, and it's coiled up and makes these structures chromosomes. In humans, we have 23 pairs in all cells apart from gametes. And they make chromosomes and they come in nucleuses. If we're talking about prokaryotes, let's talk about bacteria, slime, capsule, and cell wall, flagellum. They have 2 types of DNA. So they have plasmid and chromosomal. Not the same as the human structure of DNA particularly, but it works in a very similar way. So prokaryotic, no nucleus, and they're just found in the cytoplasm. But these are the main structures that you need to know more on the structure, and this links in with your chemistry. So you need to know that the sugar phosphate backbone is made up of a phosphate, a pentose sugar, and then a base. And these are called nucleotides. And these nucleotides, there are 4 types, and these are the monomers of DNA. And the 4 types, as we said before, A, T, C, and G. and we have an ester link and a glycosidic bond that join these. And you sort of need to know about ester links and glycosidic bonds when we talk about making polymers and polyesters and organic chemistry. But in link to the chemistry, DNA is an example of natural polymer that has 4 monomers: A, T, C, and G. I forgot to mention the brilliant humans responsible for our knowledge of DNA. Watson and Crick came up with the actual structure so we could physically see what it looked like, but they had the help of a brilliant woman called Rosalind Franklin, who used X-ray crystallography to show that it was that kind of twisty double helix shape. Worth noting, she did not get a Nobel Prize because woman. Good thing. We value and respect this these days. Also worth noting about the Human Genome Project, worth looking that up. So that is when we know all of the bases in all of an organism. So we know all of the bases that should appear in a human, and we can use that to match against other traits, diseases, and then we can look at personalising medication for people. It's pretty amazing. Huge global scientific effort. I love those. See you later. Bye.